Hello, and thank you for taking the time today for this update on StemBioSys. We really appreciate you taking time out of your very busy schedule to join us for the next 15 minutes or so to hear about our company. I would like to begin my discussion with a quick snapshot of our founding premise. We believe that traditional cell culture methods represent a compromise. Traditionally, we began with tissue, then we isolated cells, and finally we put those cells onto a very unnatural environment, such as tissue culture plastic. I think we could all agree that tissue culture plastic is a very different environment than cells experience when they're inside the human body. In contrast, StemBioSys seeks to replicate in an in vitro laboratory setting the natural environment that cells experience in the body. How do we do this? Perhaps the easiest way to explain our technology is to share with you how we make it. We begin with bone marrow mesenchymal stem cells, or other cells depending upon the matrix we're producing. We add media and growth factors to those cells. Those cells expand in number and lay down an extracellular matrix, or as the layman in me likes to think of it, they build themselves a home. At the conclusion of that process, we gently remove the cells and the fluids, and what we have left is the cell-bone matrix. So why are we so excited about this product? We'll begin by showing you what a researcher experiences when he or she puts mesenchymal stem cells on our matrix for use in their research. On the left-hand side of this image and the left-hand side of this slide is an image of bone marrow mesenchymal stem cells on our matrix, and on the right is an image of bone marrow MSCs from the same donor on tissue culture plastic. It doesn't take a cellular biologist to see that something very different is going on here on our matrix. On our matrix, we see more cells, and we see the cells aligning with one another as opposed to the random pattern we see here on the right. We can also put numbers to these differences. Here we show 3 sets of data comparing our cell-bone matrix to other technologies. Again, this is a bone marrow mesenchymal stem cell example. First, on the top left, we can see that our matrix produces more cells than other technologies. On the top right, we can see that our matrix produces cells that are smaller in size. Smaller size stem cells is a generally accepted indicator of higher potency mesenchymal stem cells. Finally, on the bottom of this slide, we looked at one gene marker we believe is another important indicator of cell potency. As you can see, there is a significantly higher expression of this marker, that is stage-specific embryonic antigen 4, or SSEA4, in cells expanded on our matrix than on other technologies. This specific data shown is from the Langer-Anderson labs at MIT, but similar data has also been seen in many other academic and commercial labs. Additionally, we have seen similar findings for other cell types. In 2020, we are pursuing several avenues to drive value. This year specifically, we have 3 strategic thrusts that we are pursuing to drive value for the company. First, we are continuing to build a life science tool business focused on working with pharmaceutical drug companies in their drug discovery programs. Second, we have identified a very promising application involving preclinical cardiac drug safety testing. And finally, we have early data on a potential therapeutic application using a new version of our matrix technology. I will touch on each of these in turn. Let's begin with our life science tool business. We have and continue to benefit from a growing trend in drug development. Researchers globally are moving away from animal models and toward in vitro human-based models. There are a number of reasons for this, including the ones I've listed here. These are ethical considerations involving animals, the potential to have better disease and safety models, and finally, there are cost advantages in many in vitro models relative to animal models. Our technology, which remember, was developed to deliver a better in vitro environment for cell culture, provides researchers with cells that give them superior tools for use in their early stage drug discovery work. Why do we know this? Our customers tell us this. Today, we have cells and matrix in drug discovery programs all around the world. As one example, at just one customer, we are involved in 4 different drug discovery programs. Our small but growing product portfolio offers researchers a broad range of platforms for use in their work. Our first product was the Salvo matrix. We use an animal product, fetal bovine serum, to make this product. For our second product, we removed the fetal bovine serum and replaced it with human platelet lysate. Salvo Xeno-free, or XF matrix, is functionally similar to our standard FBS product, and it provides a good solution for those researchers who want to do work in a xeno-free environment. We have several cell products as well. These include a range of xeno-free mesenchymal stem cells, endothelial cells, and a broad range of chondrocytes. The chondrocytes are from donors from various age groups and include both healthy donors and donors with osteoarthritis for a variety of uses. We look forward to offering more products as we move into 2021. Finally, it's important to talk about our channel. Without a channel to the market, even the best products in the world won't get sold. We have established 2 different channels to market. First, we have put in place our own sales and marketing organization to support direct sales to our customers. Second, we have partnered with a number of distributors here in the United States, as well as around the world. These distributors provide access to customers we might not be able to get to due to geographical constraints, for example, internationally, or contractual constraints, for example, as we see often here in the United States. I now want to turn to our second strategic area for 2020. We all know that drugs are required to undergo multiple safety tests before they enter clinical trials. One of these required tests is to determine cardiac safety, specifically to determine whether a given drug might cause cardiac arrhythmias when they're given to patients. Current testing protocols are good, but they're certainly not perfect. About 8 years ago, the FDA recognized this and established the SIPA committee. SIPA stands for Cardiac In Vitro Proarrhythmia Assay, in case you were wondering. The committee was charged specifically with identifying new testing protocols around determining cardiac arrhythmia profiles of new drugs. The committee, after doing work for years, concluded that IPS, or induced pluripotent stem cell derived cardiomyocytes, could be an appropriate assay for a new testing approach. However, there's one problem. Today's technologies do not enable IPS-derived cardiomyocytes to achieve a mature phenotype. In effect, they get stuck at a fetal or infant phenotype. This is shown on the pictures on the right. At the top, we have a picture of a mature adult cardiomyocyte, and at the bottom, we show cardiomyocytes derived from IPS cells. The cardiomyocytes on the bottom are stuck in an infant phenotype. This difference not only manifests itself in how they look, but importantly on the function of the cells. Again, let's remind ourselves what adult mature cardiomyocytes look like. In late 2017, we at StemBioSys began working with Dr. Todd Herron at the University of Michigan. Todd, interestingly enough, was a member and is a member of the SIPA committee and its successor, the HESI committee with the FDA. We shared with Todd a new version of the matrix that we were working on at the time. Todd compared this new matrix to his traditional approach to working with cardiomyocytes. Working with his traditional approach, Todd saw what I showed you on the previous slide, round cells that look very different than the mature cardiomyocytes at the top. Dr. Herron then put IPS-derived cardiomyocytes on our new matrix, and in just 7 days, this is what he saw. He saw a mature phenotype. Subsequent studies confirmed that cells matured on our matrix are functionally mature cardiomyocytes. Additional tests confirmed a full range of mature cardiomyocytes of these cells, mature characteristics of these cells. Finally, it is important to note that as of today, multiple companies or other non-academic organizations have validated the mature appearance of these cells. One leading company in this field has also confirmed that functionally these cells are in fact mature cardiomyocytes. Additional work is going on within these groups and with Dr. Heron. We anticipate a paper in the near future and other announcements in the months ahead. We are very excited about what we've seen with these cells. Both our industry partners and the FDA have expressed real interest in these findings. To fully take advantage of this discovery, earlier this year we announced that we had joined forces with Cartex. Cartex is a company founded by Dr. Heron and his colleague, Dr. Jose Jalife, also at the University of Michigan. Combining StemBioSys' know-how in matrix technology, cell manufacturing, and our sales and marketing capabilities, together with Cartex's world-class expertise in cardiac toxicity research, gives us a great platform for future success. One piece of evidence on what we can do together was our successful phase 2 SBIR grant that was awarded in April of 2019. This 2-year, $1.3 million grant is supporting further research on this discovery, as well as enabling us to acquire and develop visualization and manufacturing technology that will move us well along the path to commercialization. Finally, I'll briefly discuss a potential therapeutic application of our technologies. Working with the New Jersey Center for Biomaterials at Rutgers, we developed a new version of our matrix that is focused on applications using chondrocytes. In this first set of pictures, we compare the presence of collagen 2 to collagen 1, and collagen 2 being the green, on cells cultured on tissue culture plastic, on our bone marrow matrix, and in our new innovative chondrocyte matrix. As you can see, you see higher levels of collagen 2 to collagen 1 on the chondrocyte matrix relative to the other two. Similarly, when we look at just sheer numbers, we see more cells and we see faster expansion than we do on the chondrocyte matrix than we do on the other two matrices. And in fact, this might lead to a number of potential therapeutic applications involving cartilage and other tissues. We are very excited about this discovery, and we are pursuing grant and other partnerships to develop this further. I hope you enjoyed this brief overview today. If you have any questions or would like to learn more about the company or our technologies, please don't hesitate to contact me. Thank you again for your time today. I really appreciate it.